Hey, good morning, everybody. I'm hoping, it's very hard to tell if we're still gathering or not. My clock says we're right at 9 o'clock. I'm going to give folks just a couple of minutes if people are trying to get settled in and figure out the links. And then we will actually get started with what's hopefully going to be an exciting yet unique day. It will definitely be a first as far as the symposium. We're moving into, or this is our 16th year, actually hosting this research symposium, but it is the first time we have ever attempted something like this virtually. We have around 300 folks registered, even though it's a virtual event. We have close to 80 students and faculty presenting today. The actual printed proceedings for the conference ended up being over a 300-page volume, and you all will receive links to the publication. So I think for both starting and ending the day, I have to say thank you. Thank you to everybody involved. It took so many people to give this a try. Our keynote speaker that we'll introduce shortly, our speaker who will close the day. We have 25 judges circulating around the sessions today who are going to give our students feedback on their work. We have 20 session chairs and moderators trying to keep our oral sessions and our poster sessions running today. Without all of you, participants included, this just would not be possible. So thank you so very much. With that being said, I have a kind of fun opportunity, right? But it makes me slightly nervous because with our keynote speakers, typically we're bringing in folks where you can't question their professional accolades. And Dr. Gaines will go through kind of her path to where she's at. But for me, it's a bit humbling because I get to introduce somebody that I've looked up to for a very long time. One thing that is so striking about Dr. Gaines is her energy, her commitment to what she does. She sort of epitomizes lifelong learning, which hopefully that's what we all strive for. She sort of teaches us a bit about pursuing what we love. But even more importantly, I think to me, is how at the center of everything she does, humans, and caring about humans and caring about her community. And I'm not sure how to convey how much I respect that. And that starts, every facet of her world, including something very near and dear to this event, is how much she cares about students. And so with that being said, I would like to turn this over to Dr. Karen Gaines and thank her so very much for taking the time to be with us today.
All right. Thank you. Can you hear me?
I can.
All right. I'm guessing y'all can hear me out there. Well, that was a humbling introduction. Thank you so much, Dr. Pilgrim. This has been actually the most I've ever prepared for a keynote address because I think I was asked to do this over a year ago, right? Because we got canceled. It must be a year and a half that Dr. Pilgrim contacted me and asked me to do this. I was like, absolutely, the great state of South Carolina. I can't wait. I can't wait to go back, and I'm virtually back. So that's fantastic. And then of course it was canceled, and we were all very disappointed. But I am so happy to be here today. So I'm the dean of the College of Arts and Sciences at Embry-Riddle Aeronautical University. And I'm going to guess some of you out there have never heard of Embry-Riddle Aeronautical University, and that's okay, because when I was your age, I didn't hear of it. I'd never heard of it either. But it's the best job I've ever had. And being a dean has been the best job I've ever had. And what's so funny is, like, when you're in kindergarten, do you ever think about, like, what do I want to be when I grow up? I want to be a dean? No, not so much. Most people don't even know what a dean does. I'm still trying to figure that out. But the title of my talk, From Alligators to Astronauts, now that's cool, right? So when you're a kid, 5 years old, I want to be an astronaut. That's pretty cool, right? And And alligators are pretty cool. What about radioactive alligators? That's really cool. That makes you super cool. So I want to talk about alligators and astronauts because I've worked with both. I'm not really an alligator biologist, but I've worked with alligators. So I'm going to talk a little bit about that. And it was the first project that I ever worked on professionally. So it really launched me, so to speak. And I'm not an astronaut, but I've worked with astronauts. So, and that's what I do a lot today. So alligators to astronauts and what I've learned, what we've learned, and how hopefully we can apply all that to humankind. So I got to do that in like 45 minutes. So let's get going. Let me tell you a little bit about my background. This is where I'm from. Now that's not necessarily what my front yard looked like, but I am from New York City. I'm a kid in New York City in the 1970s, and this is what it looked like. The top left, that's the garbage strike of 1978. I remember it. I remember the smell, along which there was actually a school bus strike, and I went to school at a time where and it's still like that right today. Zip code kind of segregates you, and my area where I lived in Queens, grew up in Queens, was white, blue collar. There weren't a lot of African Americans, and that's when they started busing kids to school. And so there were a lot of African American kids who were bused to my school. And I just remember, my gosh, I had so much respect for those individuals. To come all the way from a different area to my school. But I remember the school bus strike and during the garbage strike, and these kids, I mean, I'm talking about when I was 7, would get on a bus, mass transit, or get on a subway and come to my neighborhood. And I just thought that was so scary. And I can imagine it was for them. And in terms of equity and disparity, during that time, my mom had a car and she could drive me to school and drop me off. But those kids had to take mass transit in the 70s, which was pretty darn dangerous. Pollution was pretty bad in the 1970s, and that started a lot of what we've done in the laws that protect human health and the environment. Clean Water Act, Clean Air Act, that really all prompted out of this era. And you see that kid on the left. Well, that's not me, but it's a scene that I recall very, very well in my childhood. I was driving over that bridge, or a bridge similar to that when I was a kid. My father was driving. And there was some African American men, and they were fishing. And you can see on the bottom of this slide, I hope it says, "Are they going to eat that?" I said that to my dad. I said, "Are they going to eat that?" Someone was pulling fish out of a river that looked just like that. And my dad said, "Yeah, they probably will." And I thought to myself, I mean, I'm a kid. I'm around like 7 or 8 years old, and I'm thinking, well, I know that's bad. They have to know that's bad, and I never forgot it. I never forgot it, and I can look back at that moment, and it had a lot of influence on what I've done in my life. So let me tell you a little bit of background about me, and I do a lot of mapping. I've done a lot of GIS work and GPS work back in the day, so I love maps. I love making maps. So I gave you a little background on my start in New York City. My father went to college. But my mother didn't. She's an immigrant from Germany, so I'm sort of first gen, because by the time I started applying to college, my parents were no longer together, and my mother, being an immigrant, didn't really know that process of how to apply to college or how to help me. So I would consider myself somewhat quasi first generation. The expectation, the culture of going to college was in my household, but how to do it was very, very foreign to me. I was a good athlete, and I was pretty good at school. And so I didn't really know where I wanted to go to school and what I wanted to do. And so the University of Massachusetts invited me to be a walk-on for field hockey. I was a goalie. Actually, my love is soccer, but that's a totally different story. But so I went there, and I worked out with the team, and I decided I didn't really love that sport, and I didn't really want to do that. But I was a philosophy major when I got to UMass, and because I guess people ask me, that's kind of weird. Why a philosophy major? And I think the answer is because I like to ask why. And maybe that went down that road of being a scientist. But you see this title, it says background as a map, when in Rome. Well, I thought it was pretty worldly, right? Coming from New York City, going to a school. My mother's from Germany. I spent time in Germany as a kid. I'd been to Canada, England, a few other countries, and so I thought I was ready. And I got to Massachusetts, and I found out they didn't like people from New York City. I couldn't believe it. There were people. Wait, I thought everybody thought people from New York City were cool. There's songs about it, movies, right? So that was my first little lesson in I don't care where you're from, right? So UMass was a very great learning experience for me. But as I was going on this journey to kind of figure out what I wanted to do with my life, I met somebody and we were just talking, and I asked him what his major was, and he wanted to be a vet, but he said, but I'm minoring in wildlife science. And I said, what? Wildlife science? Like you can get paid for that? And I thought, oh my gosh, that was it. So I went to the library. This was before Barron's, the internet, and there was things called Barron's books. And I looked up in this Barron book, like where I could go to major in wildlife biology and like what it was and all that. And so if I wasn't going to play sports, I really couldn't afford to go to UMass anymore. And I went to my advisor, and he said, well, why don't you're from New York, why don't you transfer to the College of Environmental Science and Forestry? And I had no idea what that was. Well, it's a school in upstate New York as part of the SUNY system, and it specializes in the environmental sciences. And that was it. I transferred. Meanwhile, my mom also opened up one of those Barron's books and looked at the average starting salary of a wildlife scientist and wasn't too pleased. But she didn't say anything. She's always been really, really supportive of me. So it was a whole new world to me. I got my degree in wildlife science. I got to teach field ornithology in the Adirondacks. And early on in my career, I was very interested in birds. And if you see my publications, I've done a lot of work with birds, a lot of different wildlife species, but that's where it all began. And so I went straight into my master's. That's when technology was really booming. GIS and GPS just started. I mean, we have phones here, right? And I don't know if you can see me, but the phone, the GPS in my phone is way better than the GPS that was there back in the day in the early 90s. And you needed to have a key from the Department of Defense to unscramble your location. And so I went to Purdue. And I took a lot of courses in technology and engineering there, and that's where I got my master's. But also back to this culturalism and learning about your country and learning about the environment. I'd never been in the Midwest, and if you've never been in the Midwest, it's vast. It's the farming, the monoculture, how the ecosystems have been changed. If you haven't been there, it really, from the state of Ohio all the way out to the Rocky Mountains, have been farmed. Everything has changed, and it set me on a trajectory later on in my career, and I consulted with the EPA quite a bit on agricultural runoff. I've been working with the EPA in those regards for about the last 20 years and looking at deadly algal blooms in the Gulf of Mexico. So after Purdue, that's where the alligators start coming in, and I'll get way more into that in a little bit. I got a job at the Savannah River Ecology Lab because I had that skill set, that technology skill set of mapping. And that's what they were interested in, mapping waste sites, mapping where wildlife occurred. And I'll talk a lot more about that throughout my talk. And so a lot of what I was doing when I got down to South Carolina, again, that was very, very different than living in New York City. It was fantastic. I absolutely loved it. So different, so welcoming. I consider South Carolina my home. I was working quite a bit, not only in GIS and GPS, but I started learning toxicology hands-on. radiation biology and radioecology. And what I also became was a hunter and a subsistence hunter almost, for the most part. And, you know, back to that picture that I showed you of the kid looking at the river and are you going to eat that? Same sort of thing was happening. And I saw people who were living off the land who were at risk in terms of what they were eating, where they were living, the water they were drinking. And so I was very interested in bringing all of that together. And that's when I went back for my PhD, and I got my PhD in public health at the University of South Carolina. Later on, because I never really had that deep foundation in the social science and global health delivery. Later on, I did some postgraduate work through Harvard School of Public Health and have a graduate certificate in global health delivery. I spent about 10 years at the Savannah River site as a researcher, but I love to teach. And I met my husband there, and we were lucky enough to find academic appointments. We went from South Carolina to South Dakota. So that was a pretty big culture shock, very hot, very cold, but just everything. So we went there, that was great, but a little bit isolated. And we had colleagues at Eastern Illinois University, and we found our home there. We went there. I worked my way up to chair. And I had the privilege, and it's a double-edged sword sometimes, of working with my husband since the day I met him. And so he was faculty as well. And then one day, Embry-Riddle called. It was a Friday. I'll never forget this. Embry-Riddle calls me and said, "Would you be interested in this deanship?" And I said, "Okay, did you get the right Karen Games? I'm not an astronaut. I don't fly." And they said, "But you do extreme environments." And I said, "Well, I do." And they wanted to bring a lot more life science, chemistry, extreme environments into what they do and what they do in their college. And I could bring that. And it was a fantastic journey. And I've been here for the last 5 years, and I've been able to work with some of the most fantastic people. So that's a little bit about me. I hope I didn't go too long on that. All right, so let's get back to this alligator thing. So my first day at the Savannah River Ecology Lab, I met this guy, and his name was Cub. And I told Cub I was from New York City. And he was, New York City. And we went out into the field. And to this day, I'm going to argue with you, Cub, if you're out there, your colleagues are out there. I know some people who know Cub are watching this. Cub, I fell into the swamp that day because you pushed my canoe onto a cypress knee. He thinks I just didn't know how to use a canoe, but I was pretty good at it. But anyway, I survived. Cub, I'm still here on this earth because of Cub, because he did many times keep me out of trouble. First day at work, he said to me, just don't put your hands where you can't see them. That's how you'll survive. That's me on the left. That's Cub in the middle. And that's Gator Boy on the right, on that picture there on the airboat. And Gator Boy was my first summer student for the NSF REU program. And the reason why we call him Gator Boy is because I was actually, I was doing some work on gators. My boss at the time came in and said, "We're doing more gator work this summer." And I had a grant to start working on clapper rails. And I said, I need to get a student who can get out there and catch gators, and that isn't going to kill him or herself. And that was Jay Cumby, Gator Boy. Well, Gator Boy, he's there right on the bottom there with the big beard. He's now the godfather of my children. So that's how close you become with your students. The same talk that I'm giving right now, I got to give at SREL a few years ago. So that's him making a funny face. The young lady on the right, another student of mine, that's when I was working with the HBCU summer field program at the Savannah River site. She was fantastic. She is now a vet. She went to Tuskegee Vet School, and she was fearless as well. So so my lineage to alligators still lives on through my students. All right. One last thing I wanted to say about alligators is that they're great species to study extreme environments. I'm going to get to that in a little bit. All right. Well, I'm an ecologist. That's how I started. And I still am an ecologist. That's how I look at the world. And I will tell you, so do the people who look up there in outer space. They are deeply embedded in the environmental sciences and care as much for this earth as we do as ecologists. Let me read you this passage from Aldo Leopold. It's near and dear to my heart, and I teach this passage in the courses that I've taught over the years. One of the penalties of an ecological education is that one lives alone in a world of wounds. Much of the damage inflicted on land is quite invisible to the layman. An ecologist must either harden his shell and make believe that the consequences of science are none of his business, or he must be the doctor who sees the marks of death in a community that believes itself well and does not want to be told otherwise. Back then, most ecologists were men, hence the his and the he, but you get the picture. When we're scientists, when we're social scientists, when we're artists, we see those who are educated and open their minds, we see the world differently, and that's okay. And it's our responsibility to keep on keeping on, and that's what I tell my students. There's so much hope, but at the same time, ignorance is bliss, but knowledge is power. All right, technology. Oh God, as an ecologist, I remember I kind of left New York City thinking to myself, I just don't want to ever return to that. I want to be out in the wilderness. I don't like technology. And because it's really been the bane of all the negative things that maybe have happened on this earth. So
I just want to say one word to you. Just one word. Yes, sir. Are you listening? Yes, sir, yeah. Plastics.
Let me play that one more time for you. It's really important. This was a movie called The Graduate, and it was in 1967, and plastics were just emerging. And so this man, who's the father of Dustin Hoffman's love interest, and he's telling him what he should invest in, right, which was plastics.
I just want to say one word to you. Just one word. Yes, sir. Are you listening? Yes, sir, yeah. Plastics.
All right, so plastics, right? Plastics, there weren't a lot of plastics around in 1967. It was a future. And we needed technology to make plastics. Well, it got us into this mess. 53 years later, this is what our world looks like. Plastics are everywhere. We can recycle them, but we haven't been able to keep up with that. And there's an economic consequence of recycling plastics, and many of you know that we don't do it efficiently. In some areas of the world, they just stopped recycling them. So we have a mess. We didn't really have a good crystal ball. We thought, man, this is great. Let's do this. There's so many things plastics can give us to help humankind. It's helped medicine. It's helped so many different things. But it created a mess. It not just created a mess, it's everywhere, nanoparticles in the oceans, what we find near and dear to ourselves. It's everywhere. Yep, technology got us into this mess. So is it going to get us out? I think it can. I have a lot of hope about everything. That's how I get up in the morning every single day. So as a scientist, so I started out as an ecologist. I've done a lot of different things. So when people ask me what I do, who I am, and I just say I'm a scientist. And I think if you are somebody who aspires to be a scientist, or everybody who's attending this conference today, I challenge you that you need to understand technology's role in the problem. It can cause the problem, but it's a role. It's a role that's anthropogenic, right? We're the ones that are driving that. Another role as a scientist is to collaborate, right? My days at SREL, that was the best as a researcher because we had one mission, and that was cleaning up a lot of mess. But I got to, go out of my office and I would see a biogeochemist or an organic chemist or a physicist, and we all worked together to tackle a problem. And academia, I would have to say, is actually a little bit different. There's a lot of silos, but it's really important for us to collaborate, not just as scientists, but multidisciplinary, and to be able to seek diversity in that. When you understand another person's perspective on things, it will enlighten your own. This is sort of my virtual Mount Rushmore. Think of these 4 scientists right here and their impact on this world. If you don't like calculus, you can blame Sir Isaac Newton there. He's the 3rd person there. I believe, if the story is correct, he came up with calculus during a pandemic. So I wonder what you all been doing since March. I hope you came up with something good, and I know you have. Zoom, just Zoom alone, has been a life changer, right? And then you see the perspective of Charles Darwin, right? And I'll talk a lot about that through as I keep on going here. Marie Curie, inspirational to me as a female scientist, 2 Nobel Prizes, but in terms of what her discovery about radioactive elements. And then Einstein, I think he's a really great example of when we understand our role in the problem. And Einstein was always linked to the Manhattan Project. The Manhattan Project is nuclear weapons creation in the United States. And a lot of his early work led us to the fission process. But Einstein didn't participate in the Manhattan Project. But I think that if we kind of go back to my philosophy self, if we think about Einstein and what he did throughout that time period, he wrote a letter to Roosevelt at the time warning him about the Nazis trying to make nuclear weapons, which is why then we started the Manhattan Project. So it's so hard to have that crystal ball to understand that all actions have consequences. And that's a lot of what I hope you take from this talk today. All right, so extreme environments, that's what I do. That's what I've done pretty much my entire professional life. And so if I look at this from the perspective as an ecologist, you might have heard the word niche. What is your role? What is your role? Okay, and so we feel comfortable doing certain things. And then when we're under stress, right, our niche breadth expands. And if we are resilient in that breadth, we will survive. But if we have to be kept out into that stress mode, into that, 2 over there on the x-axis too long, there'll be consequences to that. We can only be so resilient for so long, whether that is an individual, a community, or an ecosystem. Okay. So I'm going to use that thought process to give you some examples about what I do, and that's radiation and human health. And so I started, like I said, with alligators, and I work with astronauts. And the theme throughout my career in extreme environments has mostly been radiation. At least those are the examples I'm going to give today. So let me give you just a quick reminder of all this radiation stuff. I just want you to remember what radioactivity is, right? So a radioactive isotope is transforming from something that's unstable, and it's trying to go towards something stable, and it's disintegrating. And you know stuff, we've heard the word half-life and that sort of stuff, but that's it. If it's a radioactive isotope. There's ionizing radiation as well, and that's out in the sun, and we'll talk about that later on. So if we have a radioactive isotope, you might absorb something. You might get an absorbed dose. So a lot of times we worry about whether you're going to absorb something. That's the energy that will go into you. My organic chemistry teacher used to say this, and I used to say when I taught toxicology is, if you put that on your peanut butter sandwich, you might die, right? So that's kind of like your absorbed dose, because there's elements out there like radiocesium. It acts like potassium in the environment. Radioactive iodine, your body doesn't know whether you're absorbing radioactive or stable iodine. Strontium. Radiostrontium acts like calcium in the environment, so it can get into your bones. So think of that as like your absorbed dose, and we can look at that in terms of what we call a dose equivalent. We try to take that and make it into a biological effect. So and we call that a REM or a sievert. All right, and then we talk about exposure. And a lot of times that's what you're thinking about, like maybe if you get an X-ray, that's an exposure. That's ionizing radiation. It's going through you. It's not on you. Same thing with the sun. Okay, and so that's energy. And we use a unit, and I won't talk too much about that, but it's called a roentgen. I just like to say it, roentgen. I probably said it wrong. Okay, so I just wanted to give you that crash course. Let's talk a little bit about radiation and human health. Back to Einstein, right? Back to the Manhattan Project. We've played around with radiation a lot, whether it's from nuclear weapons, but we also get exposed to radiation from the sun. We've evolved under radiation, okay? And I'll get to that later in my talk. We evolved, right, with radiation coming at us from the sun. All right, there's primordial radionuclides in the earth, okay, mostly radon, right, cosmic radiation, like this said. We use it in medicine, CT scans, x-rays, right? Even if you are on an airplane, you are closer to the sun, and therefore you are getting more radiation. So somebody who's a pilot, somebody who is a worker on an airplane, they get more radiation than you do on Earth, okay? And I have a little map there just in terms of what radiation is just naturally around the United States, and a unit that we call becquerels, okay? The one on the left is an example of fallout from testing of nuclear bombs in the Bikini Islands back in the 1950s and 60s, okay? All right, so back to us playing around with nuclear weapons, and then using that technology to provide energy for the world, right? Nuclear fission and nuclear power plants, and pitched as clean energy, but we know that there's a lot of cleanup associated with both nuclear energy and us trying to make nuclear weapons. Okay, so what I want to show you here is, like I said, in 1951, the Manhattan Project was going on in Nevada, and you can see that on the left-hand side. So in 1951, you see the external internal dose from red bone marrow cancer for people who were born in 1951, and then the map on the right is the total external and internal dose of thyroid cancers in adults in 1951. So the bone marrow cancer most likely caused by radiocesium as potassium, but also strontium, calcium in the bones, and then iodine. As you can see from the example on the bottom there is the iodine lands, you know, sort of from fallout. The cow drinks it or eats the grass, and then the kid drinks it, and it can get into the thyroid because your thyroid doesn't know any different. There was a radiation emission in Pennsylvania called 3 Mile Island. In that case, there was a slight meltdown. What was happening was that the cooling water didn't go into the towers, and it emitted radioactive material. It's still debatable how many people were radiated and what those effects were. As somebody who has a PhD in public health, the epidemiology can be really, really tough. Somebody who's a risk assessor, that's what I do as well. We're trying to figure out what the cancer rate above background is. Well, as everything starts to cause cancer, it's really hard to figure out how much more, right? So because we're kind of unfortunately in this soup of toxicants that cause cancer. So cancer also shouldn't be your final endpoint. Wouldn't you like to know what's going wrong with you before you get cancer? So we've been getting better at that. All right. The next one is the Chernobyl disaster. I'm going to click off this slide real quick because it's locked up on me before, so I don't want to have that happen right now. So Chernobyl happened in 1986. The researchers at Savannah River Ecology Lab worked quite a bit with Chernobyl scientists. Actually, my husband went to Chernobyl in the early 90s. He's one of the first scientists that went over there because the Savannah River Ecology Lab knew a lot about ecology and what might happen in ecosystems and how that might affect human health. Chernobyl is sort of this unfortunate great experiment. Again, same sort of thing happened in Chernobyl. In this case, they were supposed to run a test, and there were certain alarms off. And so what happened was there was this meltdown because they kept on trying to raise some energy. And then eventually it was a steam explosion. And it was the steam explosion that pushed all the radioactive material around Europe. And I worked a little bit on that in my later years as well, where I did the risk assessment for global fallout in different areas around Europe based on prevailing winds and the geography. Again, you can see what had happened. It was in the spring, and there's something called my German grandmother used to call it the Föhn, Karen. And so the Föhn is, you can see that the explosion happened, but then the prevailing winds from the spring pushed it up north and then circled back down and deposed on all of Europe. But what's really interesting about this after a certain amount of time was that the zone, as we call it, has become an improbable sanctuary for the most elusive fauna. And there are far more wildlife species and densities of animals in the zone than before the accident. And although toxicants can really do mayhem on wildlife populations, it's not predictable. And that inflection point between habitat destruction caused by humans and toxicants that are caused by humans, they're really unpredictable on how that's going to influence wildlife populations. In terms of us, well, thyroid cancers got pushed up, as you can imagine, for the reasons why I described earlier, in children, adults, and adolescents. Okay, so let's talk about South Carolina. All right, so let me see. So you can hopefully see my arrow here. So you all are up there, most of you, right? And I'm guessing many of you have heard of the Savannah River site, but if you haven't, because I've had colleagues from Clemson going, Savannah River site, never heard of it. I was like, really? So let me give you a little bit of backdrop. So 1951, Manhattan Project's going on over in Nevada. Well, now we know how to make this stuff, right? We know how to explode it. We got to make it. So in Aiken and Barnwell counties, the Department of Energy, actually at the time it was the Atomic Energy Commission, pushed everybody off that land. There were people there. The town of Old Ellington. Now there's a New Ellington outside of the Savannah River site. And they started making weapons grade material for nuclear bombs. There were at the end about five different reactors going to make weapons grade material. And so, but you can see from this map, the same sort of thing like I was talking about in Chernobyl, moved everybody off. Wildlife started to flourish. Okay. The area is managed by the U.S. Forest Service, and it was also named the nation's first national environmental research park. where tracts of land were set aside to study not only nature, but the effects of these perturbations on ecosystems. The area is contaminated with radionuclides, metals, and organics, and now the primary mission is environmental cleanup in their technologies. A lot of what I did was risk assessment and trying to figure out what the consequences were of all this contamination, not only on wildlife and nature, but people living near the Savannah River site and people who might consume fish and wildlife in and around the site, and also looking at animals that might be vectors of contamination. So a bird that might get contaminated, then fly off and move that contamination. So I did that for about 10 years. Actually, I'm still connected to the SRS. So I've been still working in this realm for many, many years. All right. So as I said before, we've studied the physiological impact, human consumption, vector transport. You see on the bottom there, that's the nuclear reactor, that's our reactor, okay? And so that was pumping out weapons grade material for a really, really long time. What you can see on the map here, okay, what you can see on the map here is this is the radioactivity, and it's coming from the different reactors and the cooling reservoirs, okay? And what I want you to note here is that you don't see it in the water bodies and in and around the areas you saw before, which were forests. And why is that? Well, water acts like lead, it shields that radioactivity. So the reason why we were able to get this is these areas were drawn down at one point, and we were able to do a flyover in a plane like a 747, this was a long time ago, to get to measure these gamma rays that were coming from the ground. I'm going to talk a little bit later on about what we're doing today at Chernobyl with new technology. Okay, and as I said before, a lot of what I did over the years is then take that information and map it out to predict where contamination might be, so to be able to protect human health. And understanding how contaminants move in time and space is obviously extremely important in terms of protecting people. All right. And as I said before, well, cancer should not be our only endpoint, right? If we want to figure out how to help people, how to protect people, well, we don't want to wait until they get cancer, right? So one way to study how different contaminants might affect health of whether it is an animal or you is to look at different endpoints and to look at different types of scenarios. And from that, we need to have, we need to be able to look at things from different perspectives. So up on right, on the left there, you see a fish, and the fish, we're able to look at their genome, and they can reproduce really quickly. We can look, they're sensitive because they absorb dose. And then our friend the alligator there, I love alligators, I love reptiles because you can kill them, but they're really resilient. And they are extremely ancestral, and they can withstand really, really high levels of radionuclides. So in this case, you see the alligator eating that bass. Yes, I did fish for a living sometimes. And so this alligator is actually being fed a radioactive bass. And we would study the uptake of radiation and what we call the deparation to figure out how long radionuclides might stay in an animal's body. And that's really, really important. And that's the field of radioecology, because like, as you know, there's a half-life, right? There's a physical half-life of a radioisotope, but there's also an environmental half-life or an ecological half-life. How long does that radioactive material, any contamination, stay within the system? And how can we discover different endpoints to be able to predict that this contamination is occurring? What you see in the middle there is some work that I did with my husband looking, my husband's a geneticist, and he was looking at DNA strand breakage. That was one of the first endpoints. There's so many different physiological endpoints now that we can look at that hopefully will crystal ball and tell us what might be occurring in our future before it happens. You see on the bottom there, that's a wild hog in South Carolina. You know, they're all over the place. People hunt them, they eat them. That's me on the right. Not only do people hunt hogs, but hogs can get in and root around. That is actually some mud flats. They're slightly radioactive. And hogs had been through there and rooted it back up. So what happened is that there was the radioactivity was below the ground because of just natural attenuation, but then all that rooting around caused the radioactivity to be bioavailable again. So it's not so simple figuring out how contaminants will move in the environment. All right. So here's just another example about what's called ecological half-life. And it's really kind of funny because I argued with my husband for a very long time. This is some other work that we did together. He says there's no such thing as an ecological half-life. I'm like, of course there is. And his argument is that ecosystems are dynamic and they change, and so there's not just one set point. And I do agree with him about that. But the physical half-life of radiocesium is 30 years, right? So half of it's gone after 30 years. But in this case, our model, when we studied deer, half of the body burden went away in about 17 years. So that was quicker. What you could also see by monitoring the deer was that we saw perturbation. So here's the line of how we see this, disintegration or lowering of body burden, but you see these peaks, 1969, right? 1982, 1988. Well, in the 50s, right? So we had some nuclear weapons testing the Bikini Islands, and then boom, all of a sudden, this is from global fallout, right? I talked about 3 Mile Island. Then all of a sudden, that was in late 70s, 82, boom. Then Chernobyl, boom, right? There was actually just a paper that came out earlier this month from Fukushima, right, and talking about ecological half-life. And in that case, the ecological half-life in their system was 42 years. So just because it might be 17 years in a deer in a terrestrial system, it's going to be very, very different based on the ecosystem and the organism that you're studying. So what is it for humans, right? What is it for you on how we live our lives? All right. Radioecology meets technology. Well, like I said, now I'm at Embry-Riddle Aeronautical University. This is a quadcopter that we developed. So this is what we call a UAS, or an unmanned or unoccupied aircraft system. It can either be fixed wing or it could be vertical lift. In this case, it's vertical lift of a quadcopter. We use these, we make sensors for them. You can see here, there's one sensor, there's another sensor, and we'll get back to my days at the lab where we could only measure just a few things looking at radioactivity to protect human health. Well, now with these quadcopters, we can do a lot of different stuff. But before I get there, quadcopter meets droid. Okay, so this is Colin. This is Colin. So my first day at work here at Embry-Riddle, Colin came up to me and says, "Are you the new dean that knows biology?" And I said, I might be. And he said, "Well, I really want to learn biology. I'm an aeronautical engineer, and I want to grow spirulina on Mars." And I said, "Sounds like a plan." So I somewhat adopted Colin, and as he was finishing his undergraduate degree in aerospace engineering, he started working in my lab, growing spirulina and mimicking microgravity and radiation. And now he's still, he's doing his master's with us. But he was working in this lab. This is called SwampWorks, sponsored by NASA and Embry-Riddle. And this is building a UAS, but it gets deployed from either the space station or a spacecraft, and it's designed to land on an asteroid and mine resources. Now, technology got us into this mess. Is it going to get us out? Is this a good thing? I talk to Colin all the time. I said, Colin, is this a good thing? Is this what we should be doing? This technology, actually, let me talk about progression. This man right here, he owns Louisiana Steam paper mill, and he wants to invest his technology to use steam, yes, steam, to propel this droid. And what we do is we take little ice crystals off the moon or off an asteroid, melt it, and make it into steam, and that propels this droid, which is pretty amazing. But again, technology got us into this mess. Is this the type of technology that will get us out, or is we going to get farther into a mess? And that's what I always want people to think about as they're continuing in their fields. All right, so back to the quadcopter. So here's quadcopter. It's actually an octocopter, and this is at Chernobyl, and it's amazing. I got to Embry-Riddle and I started we have Embry-Riddle worldwide, and my colleagues there were working at Chernobyl. I was like, I worked at Chernobyl. So what we're doing with these octocopters is we're bringing them into the environment in Chernobyl. Okay, in this case, this is fixed wing. And like I said before, when the trees can sort of block out that radiation, well, what we're doing here is we're using also LIDAR, and LIDAR goes through forest canopies, like light. You know, you're in a forest and it's kind of dark because the light can't get through, right? If it's kind of open canopy, there's a lot of light. Well, it's the same sort of thing, but now we also can understand what the horizontal structure is of the forest. So if we use that and couple it with other detectors that can detect radiation, so we put on that quadcopter or octocopter a LIDAR detector as well as a gamma detector, then we can start figuring out maybe what's in the soil. So what we've done is we've taken some of the past research on looking at both burial grounds at Chernobyl or just areas that are contaminated and spotty contaminants, and knowing how much that tree is giving off, right? So this tree is giving off a certain amount of radioactivity, and we can sort of equilibrate that to how much radioactivity is in the soil. Okay, and then by using those detectors in LIDAR, we can figure out a better way of figuring out what the contamination on the ground is. And that's really, really important. Okay, so there's sort of an example of that. So the quadcopter can get that detection and send both penetrating light through as well as the gamma detection. And lo and behold, we can get an exposure map. And this exposure map is looking at the dose that a worker who might go back into the zone or a farmer that might go back into the zone would get from living there, eating there. So it's, again, a way for us to do risk assessments without putting people in harm's way. So in this case, technology is helping us get out of the mess. All right. Ecology in space. All right, I gotta go through this. I have a little bit, I don't have that much more time. Did you know that the International Space Station is a U.S. national lab, and that most of the science there, 80% of the science, has life on Earth applications? Okay, and actually most of the science up there is in some way biological, medicine, pharmaceutical. Most people don't know that. This is the space station. If you've kept up a little bit on that, there's a leak going on there right now, unfortunately. And if they can't figure out why it's leaking, they're gonna have to evacuate the space station. So you might want to watch that. All right, and this is an alum of Embry-Riddle, a friend of mine, Nicole Stott. She's an astronaut. This is when she's been up in space, I think 3 times on the shuttle mission. And she's an aerospace engineer. Back in the day, they only sent engineers up there, but now, diversity. Remember back to my earlier slide, diversity. You are always sending up biologists, biochemists, engineers, artists, right, to be able to learn from one another at the space station. There's a lot of science that's going up there. Nicole had basically what these astronauts do on the ISS is they run other scientists' experiments. So you apply to the International Space Station National Lab to send up an experiment, and that experiment is literally in a small box with instructions for the astronauts to conduct. There's actually mice colonies up there, and every astronaut, as you can see there, learns how to draw blood from themselves, because that's really, really important. Back to that resilience and niche, these astronauts are under a tremendous amount of stress. Think about their circadian rhythm. They're in microgravity, radiation, so much, and just being hyped up to be up there, and it's nerve-wracking. So can you imagine the stressors? Most people don't know that almost every astronaut, when they get up there, develops shingles. They develop herpes. All sorts of viruses re-express themselves because of the stress that they're under. Very much back to life on Earth applications. Think of COVID, right? Same sort of thing happens. The body's under stress, and then other things start pushing out. Okay, so there you can see also she's growing plant seeds to look at how plants grow without gravity or natural light. Pretty cool experiments. But Nicole, what she doesn't want you to remember her for any of those other experiments that you just saw. She wants you to remember her for this. She is the first person to paint in space. And the painting she has right there is actually from the image she saw from the space station, which is the Cape of an area around the Bahamas. And she also painted the shuttle, because that's what she got to the space station on. And what she does now, she's a little bit older, and she does the spacesuit art project. And what she does, and I've done this with her, and it's been some of the most rewarding experiences in my life, is she goes to children's hospitals and she paints with kids, and she takes those paintings and makes them into a quilt, and actually made it into a spacesuit. And so there's an astronaut actually on the space station wearing that art, which is just absolutely fantastic. And she still does that, her spacesuit art project, to get people, kids, never thinking, why me? I could never do that. Of course you can. Of course you can. All right, so back to evolution, Darwin, all this stuff, Madame Curie, right? Think about space. The origin, in this case, we're not dealing with isotopes, we're dealing with the sun. My friend, Nicola Fox, she's so fantastic. She's the head of heliophysics at NASA, and this is what she does. Well, imagine, right, evolution, right, is based on random mutations, which is in essence caused by different physical and chemical means that increase that frequency. Well, one of those is radioactivity, and ionizing radiation can have mutational effects on tissues and then the genome, right? So we study that perhaps fast evolution, just like my husband studied it at Chernobyl, we do that up in space, okay? I want to ask you a question before I end. So what if you were going up to the space station? I bet a few of you out there want to go to med school, right? Want to go to grad school, might be really interested in the microbiome, right? So how many of you are athletes and you've been taught, like, you better worry about MRSA, right? Because in the locker room or in hospitals, well, how would you approach the problem of bacterial contamination on the International Space Station. So same sort of thing, right? Some of my colleagues are like, we just have to disinfect the space station. I've disinfected my rocket. No, you haven't, right? When we get to space, we bring us, and what's in us, the microbiome. Even if you were lucky enough to get every single bacteria off that surface, we're bringing it up there, okay? And imagine, what about in your gut, right? You know you have a microbiome. How could radiation and microgravity change your microbiome? Change your microbiome. In space, okay, what happens is genes express themselves. Genes express themselves here on Earth. And as I mentioned before, we evolved with radiation. Okay, so when we get up in space, there's a lot of radiation, and it induces DNA damage, and it induces stress. And through the methylation process, different genes are expressing themselves. Well, remember, our niche is Earth. Well, now if we go up there, our niche breadth gets pushed out, and now we have to be resilient. How do we be resilient? Well, our body's ready. I'm going to turn on different genes. And if you're up there, you can study that. Genes actually turn on in space differently and might turn off differently than they do here on Earth. One more other example, my colleague here, Hugo Castillo, he studied at Los Alamos. What if I took all radiation away from, in this case, a bacteria? What would happen? Do you think the bacteria would get better? Perform better, or do you think it would perform worse? In this case, this species that he studies performs worse if there's less radiation. Why? Because it evolved with that radiation, and you take it away, it's now under stress. Same things happen. How many of you are familiar with this study, right? We had two twins, they're astronauts, right? And we sent one to the space station. And then CNN and all, Fox, whatever, they're all like, his DNA changed. No, Scott Kelly's year in space didn't mutate his DNA. What it did is the microgravity and the radiation changed gene expression. So when he came back down, his genome should be exactly like his brother's, right? Because they're twins, they're identical twins, but they weren't, because certain genes turned on and off differently when Scott was up there. But other things that if you read more about his journey up there, is that Scott, he being up there for a year, he started becoming pre-diabetic. He started to get high blood pressure. All these parameters, all these endpoints I was talking about before, he was aging up in space faster than his brother down here. Even kind of like this arthritis was happening to him, and slowly he's getting better, but he still has some of those effects. Okay, so there's a lot that we can learn about us up there to help humankind. So I've been going on and on. I could go on and on because I really, really love this stuff. So, but I'm gonna pause here because I have some questions that I'd like to ask you, the ones I had before about, hey, what would you do if, what would be your approach? What would be your worry if you were told to clean the space station and try to figure out whether maybe bacteria were going to be a problem? How would you approach that? And what do you think would happen to microbiome, or any other type of question that you might have of me? But I'm going to end here. I'm going to say, will technology get us out of this mess? I think it will. I'm a hopeful person, but I'm asking you, I'm challenging you out there as students in this research conference, is to always understand that actions have consequences. So I'm going to end this talk with a saying from my friend, my mentor, Eugene Odom, the father of ecology. I got to meet him many times before he died. Cooperation for mutual benefit, a survival strategy very common in natural systems, is one that humanity needs to emulate. And that's where I'll end. Thank you so much. And with that, if we have any time, I'd be happy to answer any questions. All right.
All right, thank you for that, for your presentation. We do have some questions coming in.
Okay.
And so the first question, there's a series of questions from Dan Norris. The first is the spectrum of ambient radiation the same on other planets?
No. It's not. So remember I showed the picture of the sun, and so it really has to do with where you are in relation to your sun, which is a star. So and I'm probably getting out of the breadth of what I know, but the bottom line is that gravity will be different depending on where you are, and so will radiation. And so what we try to do is shield ourselves. There's a lot of work going on, anything from pharmaceuticals to almost like a sunscreen to put on, to spacesuit materials. Obviously, the ISS has a lot of shielding. So hopefully that answers your question.
And then the next one is, are there types of radiation here on Earth that don't exist extraterrestrially and vice versa? And then the second question is, do you theorize that ecological half-life is different on other planets?
Okay, so first question is, absolutely there's different types of radiation, because remember I talked about radioisotopes, and so when when you see news stories about like, oh, we found, you know, carbon on another planet, right, or we found this element on another planet, well, because we're trying to see if things are like us. So different planets have different mosaics of elements, and maybe they even have elements that we don't know about. So if they're radioactive, they would absolutely be emitting radiation at different energies than here on Earth. So would that affect? So what was the second part of the question? So we have different.
Do you theorize that ecological half-life is different on other planets?
Sure. Yeah. So if there's life on other planets, which my personal view is yes, so would ecological half-life be different? Absolutely, because like I said before, the elements would be different. The ecosystem is going to be different. The dynamics, right? We see like these storms on these different planets. So can you imagine? In terms of erosion and deposition. So yeah, it'd be really, really cool. I mean, just on our own planet, ecological half-life changes. Back to the fight. It's one of the many arguments I get with my husband. So because ecological half-life is dynamic.
Okay, and along a similar topic, is there any relationship between ecological half-life and properties of sediments or locality?
Oh yeah, absolutely. So I think I answered that in the last in my last answer, is that you know, so the map I showed before with radiocesium and deer, just whether it was sandy soils or clay made a huge difference. And not just clays. What's really cool, if you like clay, hang out in South Carolina. Georgia has a lot of clay, but South Carolina has some cool clays because of the way it ages. And I learned this, again, diversity and collaboration. So my soil chemist friends taught me about the absorption and adsorption of clays, and the aging of that clay can have a big difference in terms of its biogeochemistry and its ability to hold on to that radioactive isotope. So as you can imagine, a sandy soil doesn't hold on to it, so it becomes bioavailable. But if it rains, all of that just goes through the sediment. Like if you pour, you're not going to put your lettuce in sand because it won't hold on to the water. But if everything deposes down and there's no rain, then that radioactivity can be extremely bioavailable. So it's totally dependent on topography, rain, the type of soil, the type of vegetation, what everybody's eating. So, yeah.
And then another question is, how long did it take to determine how accurate using that quad copter with the gamma meter and LiDAR?
Sure. So that it's all, a friend of mine, Chris Romanek, and Dr. Pilgrim worked with him as well. And he taught me, he said, these are indices. When we're using instrumentation, it's giving us an index that's relative to one another. So the question is, there's no truth in a measurement. It's an anthropogenic type of an index. But with that said, you want it to be consistent, and you want to have faith that you're measuring what you think you're measuring, right? So it takes a really, really long time. It took years because we went out in Chernobyl and sampled for a very, very long time. We studied how trees can be phytoremediators and how they can draw the different isotopes, and that's obviously species specific. Once you get a good idea of what's happening there and sample over time, then you can ground truth that. And once you get to the technology side, it's not as hard, because what you're looking for is it to be continuous. So you're getting the same measurement over time, and if you're getting that consistent measurement, then it's giving you the information that you want. So the ground truthing, years and years and years. The actual development of the sensor, years and years and years. But actually pulling it together, the two, only took a few years, couple, I think.
Okay.
Yeah.
The next question, any idea if there's a correlation between methylation of genes in space and the methylation or expression seen in people of color, specifically referencing the ACEs study, the adverse childhood events?
Oh, wow. Hold on, I'm going to text my husband, who's a geneticist. No, I don't know. I mean, what I can tell you is obviously people are different and we're diverse. And so, but I also have challenged, I teach clinical rotation in hospitals, and I do it from a public health perspective. And I once asked my students, I said, all right, I had them read a paper, and African Americans are more susceptible to diabetes. And I said, why is that? And one of my students said genetics. And I said, are you sure? Are you sure that it doesn't have to do with the zip code that person lives in and how they eat and what their lifestyle is? So it's back to what my husband always says to me, and I agree, is it's the G&E interaction, right? It's the genetics and the environment. So I don't know the answer to your question about whether African Americans might respond differently to radiation and the methylation process, but I would also argue that it's really hard to determine what that epigenetics are. So how those genes were originally turned on when you were in utero, if you're an African American, and then expressed throughout your early development. Okay, so it's a very, very complex question. So I gave you an answer that had more questions than maybe, definitive answers.
All right, and the next question is, are the animals in the Chernobyl zone healthy?
Excellent question. Are they healthy? Okay, so this is the way I answered that one. When I used to work at the Savannah River site, people would say, well, are all the animals dying of cancer? And I would say, no, they're not dying of cancer, but a lot of them have tumors. When we used to dissect bass, there'd be a lot of tumors. But wild animals tend to die from it's just tough to live outside, right? I mean, there's so many things going on. There's disease, there's famine. You might get eaten by a predator. So that endpoint of, let's say, cancer, you don't really see. And I guess I would also, so what is health? Okay, I'll just take it down to a biological statement. It's biological fitness is the amount of offspring you have that can survive to reproduce. So in that case, then we have many organisms that are healthy, fit. On the other hand, the radiation and other contamination has changed some of that evolutionary trajectory. They've shown it in birds in their song. They've shown it in behavior. So I health is a hard question, but it's changed. It's been resilient, and they can survive. So that's how I'll leave it there.
All right, and then our next question is, can you estimate, or do you think that Chernobyl will become a viable human habitat again?
Well, the half-life of radiocesium is 30 years. Okay, and so it was 1986 when it happened, and so we've been through a half-life or so. There's other radionuclides there; they're a little bit longer and shorter. We've moved back into the zone for some people. My friend, colleague Eric Peters, he has what we call the Chicken Kiev paper, and what he did is he looked at could you farm chickens on contaminated area, right? It's sort of like the alligator I was showing you that was eating the bass. We were looking at how much radiation could that alligator pick up, and how long would it take for that alligator to get rid of that radiation. Well, my friend Eric did the same thing with chickens. If you feed chickens on a contaminated area, how long would it take if I gave that animal now clean feed that was not radioactive? So you can do those types of things to make an area viable. And that's important because everywhere we are is contaminated. Back to the picture from my first slide. And so we have to learn how to live. We are learning how to live in contaminated areas. So how do we clean that up so that we can continue to live? So the short answer is yes. I think we'll be able to get closer and closer to the zone, but not right there. So, yep. Anything else?
We do not have any new questions yet. So I just want to remind everyone, in case it wasn't mentioned earlier, if you have a question, just type it into the live Q&A tab, and we will answer your questions. So we have a few more minutes.
I have a question for y'all. All right. All right. So this is what I said. So what would you do? What do you think is going to be the worst problem of bacterial contamination on the space station? So let me give you an example. So you're on the space station, right? You have to eat, right? So now what NASA wants to know, they want to be able to real time figure out are bacteria mutating into a way that we have to worry about astronauts' health. So they asked my husband to look at the mutation rate of bacteria. What should their protocol be? Should it be like a hospital? I'm going to go in and clean that surface down over and over again to prevent bacterial contamination? What do you think? Does anybody have a comment out there? What should I be worried about if I keep on doing that? If I keep on cleaning that surface in the cafeteria of the space station, there actually is a cafeteria in the space station. What should I worry about? Are there any comments out there? Because if there's not, I'll give the punchline.
No comments yet.
No comments. All right, come on, no one's watching me. All right, so to me, what we need to worry about is not creating another MRSA, right? So if we keep on cleaning, we're killing 99% of the bacteria on that surface, leaving the strong 1%, right? We know we've done this in hospitals, we've done this in locker rooms. So imagine that on the space station, but now we might be in microgravity. So microgravity, genes turn on and off differently, and radiation. And so those bacteria, that community that's now going to evolve on these surfaces very quickly, could become extremely nefarious, not to only us when we are on the space station, but bringing that back to Earth, right? Because if that bacteria mutates on the space station and I'm eating in the cafeteria and I get on my peanut butter sandwich and I eat it and then I come back to Earth, it's part of my microbiome now. And then you can guess, so on and so on and so on. So actions have consequences.
We also had a comment come in. Do we need to worry about biofilm formation as well?
I love biofilms, absolutely, yes. So I like to say that I study radioactive alligators, but the most interesting work that I've ever done was on radioactive biofilms. Same sort of thing that happens in hospitals, right? Biofilms. And so there are biofilms on instruments on the space station. And that's where my friend, Nicola Fox, where she was like, oh, we decontaminate everything. I was like, you can't. You can't get rid of these biofilms. And so absolutely, not only do we have biofilms on the outside of our rockets, and the reason why I keep on saying Nicola Fox is, oh my gosh, she posts on, so I got new couches yesterday. My friend Nicola posts on Facebook, she's like, I bought a new rocket, because she's sending a new rocket up to towards the sun. She's the person responsible for the Parker Solar Probe. And I say to her, I was like, listen, you're not going to get rid of these biofilms on your rockets. And now you're sending bacteria on the outside into space. And arguably, most of them might burn off, right? Because it's getting really close to the sun, but some of them won't. So yes, answer to your question, I think about biofilms all the time.
Okay, and I think for our final question, we have, what is the radius of the worst Chernobyl damage, and what is happening around Fukushima, if you know?
Ooh, I don't know. So I don't know I don't know that off the top of my head anymore. I used to, in terms of the radius of Chernobyl, but I can tell you, I mean, all of Europe was affected. When I was doing the risk assessment, first of all, the European Economic Community's limit for radiocesium in any food substance before Chernobyl was 0.6 becquerels per gram. And after Chernobyl, it was raised to 1 becquerel per gram because they couldn't find any food that was below that. Okay, and most food was still above 1 becquerel per gram. So it's still there to this day in terms of dairy products, in terms of the radius. All of Europe, Eastern Europe is still affected. In terms of Fukushima, Fukushima is interesting. They've tried to make this ice wall to limit the radiation because the reactor's the reaction's still happening. And again, it's based on the upwelling of water. But again, there's an old saying, dilution is the solution to pollution. I don't believe that, but it's an old saying. And so a lot of this radioactivity is getting diluted. But until we stop this reaction, it's going to continue. Now, will that hurt us? This is what I always say. I want to give you hope. I don't want to leave you with a bad taste in your mouth when you leave. We're here. We're here. My mother is 81. She's beating cancer. People are living longer. That is not an excuse to contaminate this earth. This is not an excuse to just allow climate change to happen. But we're here. We're resilient. Okay. But we have to understand that our actions have consequences. So that's where I'll end it.
All right. Well, thank you so much for giving us your time this morning on a Saturday. And thank you to everyone for attending Dr. Gaines' talk. We're going to give you guys an extra few minutes to figure out how to move into the breakout sessions that start in 7 minutes. So thank you.
Thank you. What a great day. What a great way to start the day. I had a fantastic time, and everybody, thank your mentors. They work so hard to get you here. So thank your mentors. I thank you. Have a great weekend, everybody.